Chapter 2 is dealing with cells, and the first lesson is entitled Cells and Life. The study of cells, you need to know that science is called cytology. Remember, a cell has a lot of cytoplasm in it. So cytology, the study of cells. Ology, remember, was the study of. There are several different scientists that are very important in the formation of what we call the cell theory. And so you need to know a little bit about these scientists. Robert Hooke, Anton van Leeuwenhoek, Matthias Schleiden, Theodor Schwann, and Rudolf Virchow. So let's look what you need. Now, you heard a little bit about Hooke and Leeuwenhoek in the last chapter with their being responsible for the introduction and the making of the first microscopes, the light microscopes. Well, Robert Hooke's cytology is really traced back to him, and the reason it's traced back to him is because he's the first person to call a cell, and he published a paper about it. So it's traced all the way back to when that paper was published. So know that Robert Hooke is the beginning, or where cytology actually began. You need to know the paper, and I might ask you the year. The paper was called Micrographia, and in it he had his drawings, and you can see there a picture of, or a diagram of, what his microscope was like. Very different than the ones that we have nowadays. And when he was looking at cork under the microscope, it looked like this on the right. Those are two drawings in Micrographia that Robert Hooke made of cork. Now, cork is living. It comes from a living tree. It's actually the bark of it, and it's a type of oak. It's called a cork tree, and it's a type of an oak. So because of these little boxes, they reminded him, like side by side, lined up, of cells in a monastery, you know, where the monks and so forth slept, or cells in a prison. And because of that, he was the one that coined the word, or the one that first called a cell a cell. And so in that paper, Micrographia, is where he wrote about it. He wrote about, and he put his drawings of the things that he looked at, mainly the cork that he was looking at. So he saw, and you need to understand this, when he was looking at the cork under the microscope, remember his microscopes only magnify 30 times. Now, our lowest power on the microscopes that we're going to be using, they magnify 40 times. So even our lowest power is higher than what Robert Hooke saw. But remember, this was back in 1665. So our microscopes, the highest that we have in magnification on our light microscopes, and they're called compound light microscopes, is 1000 power. That means it takes an image and can blow it up to 1000 times its original size. With Robert Hooke, his was magnified or blown up 30 times. He was looking at the cork. Now, the cork had been taken out of the tree, so it was no longer living. The cells weren't. So all the living stuff had decayed and gone. So he was really looking at the empty cells. Well, he saw a boundary around it, and he saw these cells again, which reminded him of cells in a prison or a monastery. So he called them a cell. What was in it, he only saw the cell wall of that plant cell. That's what he was looking at, the dead cell walls of the cork cells. Here's a picture also of his book, or the paper it was that he wrote, Micrographia, 1665. Anton van Leeuwenhoek, other than what you learned about in his microscope, remember, magnified about 270 times, he, around the same time about as Robert Hooke, was also looking at stuff under his microscope. And I'm going to have a picture of his microscope, very different than what Robert Hooke's looked like. And so he began to observe these tiny little objects with his microscope. Now here's what his microscope looked like. Nothing like what I would call a microscope. I wouldn't have even guessed that was a microscope. See that little point there? Light came through that hole, and that's what then magnified, or with the lens magnified, and the light then brightened and shone through the specimen. He looked at stuff like drops from lake water, scrapings from his teeth and gums, and water from rain gutters. Now, there's a lot of microbes, especially in like gutters. And also you have microbes living in your on your gums. So what he did then is, when he saw these magnified, remember, 270 times, he started drawing these little creatures that he saw. And they reminded him of little teeny animals, so he called them animalcules. And so you get to see some of the animalcules. I would like to know what some of those really are. Of course, you can see a little starfish and stuff like that he saw. Now, this, goes way up in time. There is 2 German scientists that are also very important in the formation of the cell theory. And this is one of them, Matthias Schleiden. He's a German botanist. So if he's a botanist, what did he deal with? What was he studying? He was studying plants. So in 1838, so that's a long time from the first publication of the micrographia in 1665, Schleiden made this very important statement. All plants are composed of cells. So make sure you know that statement with Schleiden. He was a botanist, so he's dealing with plants. And you may think, well, I've learned that since I was in elementary school. Yes, but back then they didn't know it. This was the first time anybody stated that living creatures were composed entirely of cells. And of course his living creature was what he was studying, and that is the plant cells. Now another German working at the same time, 1 year later, made a very similar statement, but he was a zoologist. And of course, what do zoologists study? They study animals. And I'm trying to get the slide to move, and it's not moving for me. Whoop. Okay, there we go. So he was a zoologist. Zoologists study animals. So he realized that all animals were composed of cells. So he made that statement a year later, 1839. And that was a very foundational principle of the cell theory. The next guy, also German, come on, having trouble with my clicker today. He is a German. He was a pathologist. That's a person that studies tissues to see if they're diseased and that kind of stuff. And his name is Rudolf Virchow. And he is referred to as the father of pathology. You've heard of pathologists. That's like if somebody has a tumor and they take a piece of that tumor, they send it to pathologists to study it to see if it's normal cells or if it is like cancerous cells. And in 1855, he made this very important statement that you need to know. New cells are formed only from cells that already exists. So he's saying cells come from living cells. A very important principle, because evolutionists have and believe, and this is part of their cell theory. What he's stating is life is coming from life. These new living cells are coming from living cells. It wasn't from a big explosion. It wasn't because, some of the evolutionary beliefs out there, some of the first living organisms were just little tiny microscopic one-celled organisms. And they said it came from nothing, out of chaos. Well, he's saying cells come from pre-existing cells, cells that are alive. So that's a very important thing that you could actually use in talking to somebody who believes evolution. Right here, one of their scientists says life is coming from life. Cells come from cells. So these scientists all led up then to what is now known as the cell theory. And there are three important basic principles of the cell theory. Make sure you can list these on the quiz. One is all living things are made of one or more cells. And of course, who did that go back to? Robert Hooke. Nope. Leeuwenhoek? Nope. The two Germans. Schleiden and Schwann. They're the ones that said all plants and all animals. And at that time, there were only two kingdoms. So all living creatures were either a plant or an animal. And they said that that's what they are. They're made of cells. It's God's building blocks. The cell is the smallest unit of life, and we talked about that. If you actually take the cell apart into the parts that it makes it up, like the nucleus, the boundary, the ribosomes, and so forth, what we're going to study in this chapter, you no longer have a living thing. The life can only be broken down into the cell and still be called life. And of course, the last statement of the last principle of the cell theory was made by Virchow. All cells come from pre-existing cells. So again, make sure you can list those on this quiz, lesson 1 quiz. And here's a picture, because a picture is worth 1,000 words, of these three. So notice they got a flower. They took a little piece of the flower out to show you that that is made of cells. And we'll be looking inside some organisms and see all the cells, some of the cells that make them up. And there's a picture of, actually a diagram of a cell, the smallest unit of life. And here they're showing the process of how a cell makes another cell. A pre-existing cell is making new cells. That process is called cell division. And it's made up of mitosis and cytokinesis, and we'll be studying that. But this is just illustrating the process of a living cell making another living cell, and then each one of those making another living cell. So it goes from 1 to 2 to 4. And 4 would go to 8, and 8 would go to 16, and on and on and on. So the key concept here: how did scientists' understanding of cells develop? And it's with those central scientists that I listed at the very beginning, from Robert Hooke all the way to Virchow. Now, the basic cell substances. When we look inside a cell, we're going to find out that most of it is water, because plasma is mostly water. I think it's about 90% water. So the main ingredient in any cell is water. A water molecule has two parts, and hopefully you studied a little bit of this last year. I know you're definitely going to study a lot more of it in 8th grade when you study physical science. But if you studied about atoms and elements and molecules, well, when you have an atom of water, it is made up of two things: hydrogen and oxygen. And it's H2O, so that means there's two hydrogens, which are pictured in the blue, and one oxygen molecule. It's interesting how two gases combine together to make a liquid. And they're talking about the charge. And these atoms can have charges, so they attract each other, the opposites. So the two hydrogen are attracted to that one oxygen molecule because it is negatively charged and they are positively charged. And it's that attraction that holds those atoms together to make what we call one molecule of water. Because a molecule is two or more atoms that are combined together. Now, water and salt, which we have both inside of us and in our cells, and also the fluid that bathes our cells, yeah, water and salt. Both have positive and negative parts. So that's how they are attracted together. And there, of course, is a picture of salt on the left, and then a picture of salt being dissolved inside the water and how they're attracted then to those water molecules. This leads up to, remember we have atoms. Atoms together, two or more of them, make a molecule. Now when you have a substance that is formed by taking a whole bunch of molecules and putting them together like in a chain, that is called a macromolecule. You do not need to know the word origin. It technically means long because it's a long chain of smaller molecules together. I look at it as macro. Macro is large. Micro is tiny, like a microscope. Microorganism. So macro is big. And I think, why are we looking at these? Because there are 4 types of macromolecules in the cells. And you need to be able to list these four, and you need to know the information about them. And these are essential to life. So we have 4 macromolecules that are essential to life. 3 of them come from food. This first one does not. They're called nucleic acids. And they are a macromolecule, remember, made up of many smaller molecules, a chain of them. You need to know what makes up a nucleic acid. And it's actually a chain of nucleotides. Now that may seem strange right now because you probably don't know what a nucleotide is, but we are going to study later on about DNA and what's in a nucleotide and how does it make up DNA. Because there's really 2 types of nucleic acid. And right now you can just use their symbol. They're called DNA and RNA. Both are nucleic acids. And we're going to learn their importance later on. Proteins. Now this is in your food. Proteins are long chains, that's why it's a macromolecule, of amino acids. So I say it like this, it's the building blocks. So if I ask you, like, what's the building blocks of a nucleic acid, you would say nucleotides. What's the building blocks of protein? Amino acids. What's the building blocks of life? A cell. Now proteins are involved in almost everything that happens in a living cell. They are vital. Well, all of these are essential to life. So what are the other 2? They're found in food as well. Lipids. Now this is why I said they're essential to life, because a lot of people think all lipids are bad. Lipids is a fancy word for fats. Lipids and fats are the same thing. So they're large macromolecules made up of fats and other things with the fat. They do not dissolve in water. That's one of the characteristics of a fat. They're not dissolved in water. Now, when you look at the types of fat, you think, I thought we're not supposed to have fat. Fat's bad for you. Yes, there is bad lipids, and there are good lipids. A good lipid, which is important to maintain your good health, is HDL. And you hear of that in cholesterol. They call it bad cholesterol and good cholesterol. HDL prevents the bad lipids from getting put or deposited in your blood vessels. And that's what clogs them up and causes hardening of the arteries. It causes strokes, causes heart attacks. That's the bad. So they're keeping the bad. So that's a good fat that you want. Fat's also healthy for your brain to work, the good fat. The bad fats are total cholesterol, LDL cholesterol, VLDL cholesterol, and probably what you may have heard of, triglycerides. And Dr. Oz, hopefully I'll remember to show that to you. He takes the man's blood and shows him the fatty layer in his blood from all the triglycerides that he has, and how he's just a heart attack waiting to happen. So I want you to realize that fats, it's not, if you have any kind of diet that totally cuts out fat, that's bad. Fats are essential. It's the bad fat that we need out, not the good fat. Or technical name, lipids. And then the other is carbohydrates. So proteins, lipids, and carbohydrates are all found in the food that we eat. Carbohydrates are important for stored energy. Lipids store energy as well. Carbohydrates provide structural support, which you'll see many of the others do as well, especially the protein. They're needed for communication. You think, well, they talk to each other? No, we'll talk about more. That's a chemical communication. They communicate to each other. And they're made of 3 different kinds of atoms: carbon, hydrogen, and oxygen. That's the CHO. The examples of carbohydrates are sugar and starches. So when you eat sugar, when you eat starch, you are eating carbohydrates. And again, there are some that are healthier for you and some that are not so healthy. Too many carbs, not good. That's what they commonly refer to as carbs. Carbs are carbohydrates. So realize that all 4 of these, nucleic acids, proteins, lipids, and carbohydrates, are essential molecules, macromolecules to life. And this last picture is showing you some of the functions that we'll be studying when we look at the cell and inside. The nucleic acids, especially the DNA, is found only in the nucleus. RNA can be found in both the cytoplasm and the nucleus. And the DNA is what contains the genetic information. Proteins, look at all they do. They communicate, they transport, they're chemical breakdown substances. We're going to learn that a lot of them are in enzymes that help our chemical processes go on inside of our cell. They're also important in building parts of a cell, building muscle tissue, and their structural support. Of course, that would be along with the muscle. Carbohydrates are for energy storage, structural support, and communication. And lipids are energy storage, protective membranes. Membranes are actually made of proteins and lipids. That's what a cell membrane is made of. And they're there for communication as well. So each type of macromolecule has a unique function in the cell, and they all 4 are essential to life.